Hello everyone, welcome to today's PTI webinar. Today we will be discussing assay development from scratch to validated assays. Today's speaker is Dr. Melody Sauerborn. Melody is a consultant and a frequent trainer for PTI. She will be teaching day one of our Analysing Bioassays and Immunoassays course on the 21st to the 22nd of February. And she's also developing an online version of the course for us, which will be running for the first time in June this year. Melody will be speaking for about 30 minutes today, with an opportunity for questions at the end. You can submit questions as they occur to you in the questions tab on your screen. I'll now hand you over to Melody.
Thank you very much. Welcome to everyone. So I will give you a brief introduction into our Analyzing Bioassay and Immunoassay two-day workshop that is normally given 3 times a year, with the next one coming up on the 21st and 22nd of February. So I will just dive in immediately, as we don't have that much time, and I had to pick some aspects to discuss with you today, and we will have 4 topics that are really one-on-one taken out of the course. So everything that, all the slides that you will see today are actually slides that will come back during the 2-day workshop, whereas I will be teaching day 1, as I am more the immuno-bioassay person, and day 2 will be presented by a dear colleague and now also friend of mine, who is more in the CMC production potency assay world. So we're actually a very good match when it comes to giving you the full overview of any possible assay development that you may need during drug development. So let's just dive right into the immuno versus the bioassay. Well, the focus in the course that we're teaching is, in the first instance, assays that are run under a GLP-GCP assessment of drug safety and efficacy, and that can be anything for pharmacokinetic, tox, or immunogenicity. So that is the bioanalytical part, and that is where my expertise lies, and that will be the focus on day 1, including validation. And then day 2 will be the GMP assessment of manufacturing processes. So there, the assays, especially potency assays, are designed to look at influence on formulation, but also stability and release of your drug then for the clinical trials, but also then, of course, post-marketing. We have the cell-based assays that are, in general, classified as functional, biochemical binding, and the major thing there is the potency. We have the immunogenicity aspect, where we need assays as well, and the immunogenicity aspect has a screening, confirmation, and titer assay that is, in general, an immunoassay. And then we also have what is called a neutralizing anti-drug antibody assay, and that is, in most cases, a cell-based assay. So from my side, you will have both the immuno and the bioassay, whereas on day 2, when we talk about the potency and the CMC, you will be fully focusing on cell-based assays. And then you have the immunoassays, and there it is the development, the validation, the sample analysis, and that could be, for example, PK or tox assay. So when we look about the ligand binding assays that are actually available on the market, and I'm sure there are actually more these days, so this is just an example, or this is just an overview of assays that I have actually worked with, or that these are projects that I was involved with that use this technology. And I always like to say that I can learn as much from the participants as I can, as you may learn from my side. So there is a number of ligand binding assays. For example, the typical ELISA, and the ELISA is the immunoligand binding assay that is used the most to demonstrate, and we will come back to that in a second. But the same principle of, immobilizing something, binding your target, and actually detecting it with an antibody is used in all of the other techniques as well that are shown here, like, for example, in alpha ELISA, and there you have beads that are bound with your receptor, so you have donor and acceptor beads. You have the RIA, the radioimmunoassay, which is a bit of an, well, one of the first assays that was around and was used, for example, for insulin back in the 80s, when insulin was one of the first products that was actually on the market, recombinant human insulin, and there are other techniques there as well. And when we talk about bioassays, cell-based assays, you have a number of different cell-based assays that, of course, are using cells, but you can look, of course, at the proliferation of cells. You can look at the apoptosis of cells. You can look at reporter proteins. So instead of looking at proliferation or apoptosis, so waiting that long, you will stimulate your cells, and then upon stimulation you will actually have a reporter protein that is used for detection. You can look at phosphorylation, which is similar to the reporter protein, because the phosphorylation of a particular target receptor is facilitated by the presence of the drug. And therefore here we don't wait until phosphorylation of the receptor actually leads to production of a protein, where there we will intervene earlier, open the cell and look at the phosphorylation status of our target receptor. Or we could look at gene expression or cytokine expression, and in that case you have what is called an ELISA spot. Well, I mean, when we talk about immuno versus bioassays, the most important aspect and the most important distinction between the immuno versus the bioassay is, of course, surprisingly or not, the cells. So I will focus now a little bit more on the cells, on the most important aspects when you actually do a bioassay. So when you start at the basis, which means that you need to have a cell line in order to run your assay. And there are multiple ways you can obtain a cell line. It could be homemade or it could be purchased from a vendor. But in any case, you need to, for yourself, you need to make sure that your cell line has a certain must-have characteristics. And for example, it should exhibit a specific response to the drug. Well, it seems obvious, but it's always good to actually write it down as well. It should have a proper sensitivity, and that is also in correlation with the proper dynamic range, because the shorter or the closer your or the steeper your dynamic range is, the more problems you may have later on to actually convert a cell-based assay into a NAB assay, or to be able to actually have a proper parallelism comparison. It should be matrix tolerable, and at the end of today we will briefly talk about matrix interference, as that can have a major effect on your assay, and you may have to add much more layers to your cell-based assay as you intentionally wanted to, and that is simply because your cell line is not matrix tolerable, and there are some components of a matrix that will actually interfere with your assay. You want to have a suitable endpoint, and the suitable endpoint is something that we briefly discussed in the previous slide. And in that sense, we do have what is called an early response endpoint. We have an intermediate and late response. The early response endpoint is, for example, phosphorylation of a receptor. The intermediate response is when we go further and we let, after phosphorylation, let the signaling cascade go on until we actually start producing some messenger RNA. So at that point we can also break open the cells and actually look with an RT-PCR what kind of mRNA is up or down regulated. Or we could go on and do the traditional cell-based assay, which is the late response, which, as we briefly saw before in the slide, is in a lot of cases proliferation of or apoptosis. And the main difference between the early, the intermediate, and the late is simply the turnover of the assay. The early response is something that can be measured up to or 1 or 2 hours after you actually added your drug to the cells. Intermediate will take a bit longer, and late responses can be up to 5 or 7 or even longer days, depending on the cell line and the response that you're looking at. The cell line characteristic, once you have seen that the cell line actually has a proper dynamic range, it can handle your drug, it will respond to your drug in a suitable endpoint that you can measure, you have to actually get your cell line ready to be used in an assay and then eventually be able to validate this assay. So there are a number of things that you need to optimize before you actually go into the validation process. So we are now in the feasibility part, and in that particular case, you want to, for example, optimize the number of cells per well. So it does make a difference if you see 20,000 cells per well or 30,000. You want to establish a maximum passage of your cells, and that can be extremely important later on for monitoring because you always need to have a control in your assay that your cells are actually responsive. Because if you don't have this particular control and you don't know exactly at what passage your cells will start to lose responsiveness, you may think that your drug is less potent, but it could be that the drug is as potent as it was before, but simply the cells are not suitable anymore. So you need to have a maximum passage of cells in order to control for the effectiveness of your cells. Incubation time may have a huge influence on the steepness of the curve, and that may then later on be a factor that can come in handy when you do parallelism testing. The use of medium and antibiotics, and later on the effect or lack thereof, as you do want to at some point work without antibiotics, but medium in itself can also have a huge impact on your cell line performance, and you should always try to get your hands on different vendors in order to compare if you are forced throughout the validation process or later on to switch your medium for whatever reason, and believe me, I've had that case, it happens. You want to make sure that your cells can tolerate switch of medium, which is essential. So if you have gone through this validation part, through the feasibility part, sorry, you will go on to validation of the cell-based assays, and due to time, I cannot go through these particular aspects, but if you look at what is the difference, for example, between a cell-based assay and the ligand-binding assay, when you look at the parameters that you have to look at throughout the validation process, for example, specificity, precision, range, robustness, stability, or the accuracy, relative accuracy, there is no difference in the validation parameters, but the execution may be different between a ligand-binding assay and a cell-based assay. And these are aspects that will be discussed in the course at length, and then of course for both immunogenicity aspects, but then later on also of course for potency aspects. When we look at the critical factors that we need to take care of in assay development, for the sake of time, I did pick the ELISA and a sandwich ELISA in order to highlight one of the aspects. So when you look at a typical sandwich ELISA, you have several steps that you have to run through, and that is, for example, the coating and the blocking. You will add your sample, you have a detection antibody, you will have a detection system, and you will have to add your enzyme in order to be able to read out your system. And each of these aspects needs specific consideration, which are all addressed, but for the sake of today, we will actually focus on something that is well, sometimes taken for granted, and these are the ELISA plates. Because most people, when they do antibodies, they take what are called high protein binding plates, but in a lot of cases, people are not actually really aware what it means. So besides all the other aspects that you have to look when you look into coating and blocking, the ELISA plate is actually something that is neglected by a lot of people, and therefore I thought I would pick this particular aspect to just demonstrate here what it actually means if you have a modified ELISA plate that is modified in order to accommodate protein binding. Well, the original ELISA plates, they are based on the passive absorbance of proteins through a number of interactions, and that could be hydrophobic or ionic interaction. So when you look at the original ELISA plate, and all of the ELISA plates are in basic polystyrene plates, they are low binding without modification. So when the drug development process expanded for biologics and especially therapeutic antibodies, people started to realize that they needed to modify the plates in order to make them better binders for antibodies. So what they did, they modified them by radiation or other modifications. And what does it mean when you do that? It actually exposes hydrophilic groups on the plate surface and therefore enhances the protein binding. So this is the reason why those, well, if you look at your ELISA plate, in a lot of cases there is like a, it is called M polystyrene. So if you really look into what is your ELISA plate made of, in a lot of cases it would say a little m and then polystyrene. And this little m is exactly what I mean with modified. That really means that your plate has been modified to be a high binder. And the theory behind this is that when you have this modified polystyrene plates, the antibodies will mainly bind through their Fc part, so thus they will stand up and actually expose the binding site towards the open face of the ELISA plate where your product will then be put onto, whereas on a non-modified plate they will bind everywhere. So in a lot of cases they will actually bind lying down and not being able to be fully accessible by the target protein that you want to detect. Another issue for ELISA plates or in any kind of assay is the possible effect of a hook effect. And that means that you think the more you coat, the more the chances that you can actually capture your protein or your target drug. That is not the case, because antibodies that you coat on your ELISA plate, they interact with each other as well. And if you have an excess of coating antibody, you can have binding amongst the antibody, the coating antibody, which will lead to steric hindrance, and you will actually lose signal of your drug with an increasing concentration. This is a demonstration of a hook effect on a coating basis, but a hook effect can happen at any point in your assay and is something that has to be considered and will also be discussed in more details. And then realize that when we do the assay development and feasibility, and a lot of times if we do potency assay, that is something different because we don't work with the drug in anything else than the actual formulation. But if you work in PK, immunogenicity, so you actually work with plasma samples or any kind of biological sample, you will at some point have to interact or introduce actual matrix, blood, plasma, serum, any type of biological matrix. And the next slide is to show you that just by introducing 5 or 10%, in this case it was non-human primate serum, you saw a clear effect on the reference curve that we were measuring this particular assay. So what you see here, the blue versus the red. So the blue is the 5% matrix containing sample, and the red is the 10%. So what you see here is that from the blue to the red, you have 50% reduction in the C value, which means that there is a clear effect of the matrix on this particular assay. And this brings me to the matrix interference, because this is something that if you have an assay that is run in biological matrix, is something that a lot of people run into, and it can be a very annoying problem. What is a matrix or analytical interference is the impact on an analyte in a sample that will hinder or falsify the correct value. It doesn't necessarily mean that you will have a negative value because it will suppress your signal. You may also end up with having a compound that will interact with your detection antibody and will actually lead to false positive results. There are two types of matrix effects, and one is a substance that has a direct impact on the analyte itself, but there are also substances having an impact on the assay reagents. So in most cases, substances that having an impact on the assay reagents, they create more false positive signals, whereas substances having an impact on the analyte normally create more false negative. But that is general, or generalizing it and expressing it from my perspective and my experience. but it can be the other way around as well. But generally there is a tendency towards false negative when you have an impact on the analyte, false positive when you have an impact on the assay reagents. And to give you an example for this matrix effect is what is called, when we look in the world of immunogenicity, is the problem of drug tolerance. And that means when you have a patient and the patient will make antibodies against the drug, and you actually want to measure the antibodies against the drug, the antibodies as they are made against the drug, and if there is drug present in the patient, the actual antibodies will bind to the drug. So why is it especially a problem with therapeutic antibodies, and why is it a problem for the assay? Well, in the presence of high drug concentration, that is the problem with therapeutic antibodies. A lot of times therapeutic antibodies have a long half-life, meaning that normally patients will receive therapeutic antibodies and they will build up the presence of the therapeutic antibodies throughout the therapeutic or the treatment with the drug. So therapeutic antibodies have the side effect that in a lot of cases anti-drug antibodies are formed and no one sees it, and that was the case actually with adalimumab, which is a human antibody, and patients had remissions with their disease, but no one could explain why. And people were really convinced that there were anti-drug antibodies against the drug, but they couldn't measure it. And that is because when the anti-drug antibody binds to the drug, because there is still a lot of drug in the bloodstream, they will not be able to bind to an ELISA plate because they're actually already bound. And so what people do when they realize that that could be an issue, because in a lot of cases you have a PK data available as well, and if you see that your drug all of a sudden has a shortened lifespan, and in a lot of cases that is because it is broken down by the immune system quicker in this immunocomplex, you should be, you have to look in how well is your anti-drug antibody assay actually drug tolerant, and that is, the definition there is the maximum amount of free drug in a study sample that allows detection of anti-drug antibody at an acceptable sensitivity. And there are, in this example, in the next slide, there are ways to actually test for this drug tolerance. And it is not that there is, when you go through validation of this particular assay, it is not that you have to have a certain limit. It is not that if you say, okay, I can detect that much ADA in the presence of this much drug, it is not that there is any requirement to it, but you should mention it that, you know, okay, my sample X has come back negative in my ADA assay, but from the PK assay, I know that I have this amount of drug, and therefore it is safe to assume that we cannot detect ADAs because if they are there, they're actually bound to the drug. So this is the rationale behind it, but there is no requirement attached to it, meaning that if you have a certain amount of drug in your patient sample and you're not able to measure the anti-drug antibody, you will not be rejected, or the assay will not be rejected. So the consequences in my assay, if you have immunocomplexes, is that you get a false negative signal because it will immediately bind the compound that you want to measure, the anti-drug antibody. But it also has a clinical effect because it will deposit complexes in tissue, because these anti-drug antibody drug complexes, especially if the drug is in a therapeutic antibody itself, can be rather big, and they actually get literally stuck in tissue, liver, you call it, and that can call to hypersensitivity type 3 responses, which will have a clinical impact on the patient. There are solutions to these immunocomplexes when you think that you have immunocomplexes present, and that is the reason why you cannot measure anti-drug antibodies in your ELISA. What you can do is you can acid disassociate your drug from your anti-drug antibodies, and then you can actually measure it in the assay. There are pros and cons to this approach, and there are more sophisticated approaches these days, which will be discussed at length at the course as well. But the main thing is that yes, you can still measure anti-drug antibodies in the presence of the drug, but you may have to do a pre-step before you actually apply it on your ELISA plate, and that also means that you have to have one extra validation step in your project built in, because you also need to show, of course, that this pre-disassociation step doesn't have a negative effect or biases the actual ADA ELISA later on. Well, there are a lot of things that you have to consider, and we have discussed, especially also for the assay development, a lot of those single influences. So we talked about the ELISA plate. We briefly discussed the concentration of the coating antibody. So doing it like this is what is called OFAT, so the old way, one factor at a time. And the OFAT is time and money intensive, and a lot of times what you do not do is to look at interactions between two factors. So it can be that it is the concentration of the coating antibody in combination with the matrix effect that will have the biggest influence on your assay, but at this point when you do it one by one, you will not immediately see that. So what people started to do is to look at not only individual influences on the assay performance, but also active influences, and that has led later on to an actual statistical tool, which is called the design of experiments. Well, the design of experiment is a planned approach for determining cause and effect relationships, and not only on single influencers, but also the combination of single influencers. Here it says the quotes, both of them from the DOE Simplified. That is a book by Anderson and Whitcomb, and I have to say that if you are relatively new or even advanced in the DOE, this book I can really recommend reading it, because it gives you a very good basis from actually really when you have never worked with the design of experiments to really actually advanced users. So I can really recommend this book. And the funny thing is that the design of experiment itself actually originated from the agricultural world, because they wanted to control and monitor the effect of changes in the culturing process, and they wanted to do that by manipulating not only one factor but multiple factors at the time, like the temperature, the culture medium, etc. And as that moved on, other fields recognized or realized that this particular design of experiment approach could actually be used for many processes. And this is how a DOE matrix could actually look like. So what you see here is the terms, so the sorted parameter estimate. So you have here serum, drug, serum alone, so that's a single influence. And what is interesting here, if you look at the probability, so at the right side of the table, you have a probability, and the first four factors have a probability of 0.001, which means that the first four terms that are mentioned here are the most influential parameters of your bioassay, which means these are the four influencers that will have the biggest effect on your bioassay if something is not going right. So this can help you to troubleshoot your multi-layered bioassay. And well, this concludes what I wanted to tell you from some of the aspects that we will discuss. And I hope you learned something from it and it intrigued your interest. So I hope I'll see you back on the 21st and 22nd of February in London. And I'm happy to answer any questions. And I think there may also be, you can always, I think, email PTI as well if you have any other questions that come up in the next few days or weeks, and I'm happy to answer them as well. And of course, I'm happy to answer all questions in London when we see each other face to face. Well, thank you very much.
Thanks, Melody. I'm just seeing if there are any questions that have come in. I can't see any there so far, so I'll just give everyone a few minutes to submit any questions they have. Folks, please do submit those questions in the question tab. So one person asks, what is your experience between assay variability and endpoint methods for CBA?
Well, there is one longer case study that we will do in the course that deals with the choice of endpoint methods, and what was seen in that particular cell-based assay was that when you do early endpoint methods versus late endpoint methods, the dynamic range was much bigger. So some of the assays couldn't even be used with an intermediate or late response because they had such a small assay window. So that is something that that can certainly influence how your assay performs. And in general, I am in favor of having an early endpoint because it gives you less variability, because you simply have less steps that a human can be involved and can lead to variability coming from human handling. And also, of course, the cells itself will be less exposed to environmental influences, incubation times, things like that.
Okay, so there don't seem to be any further questions. Thank you very much for your presentation today, Melody, and thank you to everyone who attended as well.